Russell Schwartz as the session keynote speaker, and Dr. Russell is chair of the Department of Computational Biology in CMU and has a long time experience doing computational biology. And we are fortunate to have him to talk about his research today. And welcome, Russell. You can start.
Thank you, John. Thanks for having me here. Let me just share my screen here. Okay, I hope everyone is seeing my screen now. So I'm, as you said, I'm Russell Schwartz from Carnegie Mellon University. I want to tell you today about some of our work in cancer progression risks, so in particular, bringing machine learning to understanding some of the factors that influence the risk of progression of cancers, where progression here means the process of a cancer moving from different stages of disease development. For example, it might be from a healthy cell or healthy tissue to a precancerous lesion, precancerous to cancer, and then from primary cancer to a drug-resistant or metastatic form. And what we're really interested in here is what underlies the risk of progression. So what is, in a sense, the predictive information here? So it's perhaps a bit backwards from the way we might sometimes think of knowledge-guided machine learning. We're not using knowledge to try to create better predictors per se. We're using machine learning to try to understand what knowledge leads to better predictors. But to give some background into why we're doing this, I need to tell you a little bit about what the relevant knowledge is here. And that's something that comes about from largely the idea of cancer as a disease of evolution. So cancer, when I say it is a disease of evolution, what I mean is that cancer is governed by laws of evolutionary diversification or drift and selection. Typically, we believe a cancer would begin with a single cell that acquires some kind of mutation. Very often, maybe most of the time, that mutation will be something that itself induces a hypermutability phenotype. So that is, it primes the cell to more rapidly generate mutations, and that sets the tumor on the path, or really the healthy cell at that point on the path to ultimately becoming a tumor. It leads to expansion of heterogeneity as different cell lineages are rapidly acquiring mutations from one another, becoming distinguished from one another. And this genetic heterogeneity creates the opportunity for selection. So for some combinations of mutations leading to preferential benefits to the tumor and growing more rapidly, escaping controls on cell growth, escaping treatments, moving to new sites, and so forth. So this has been known for a very long time. I cite here Nowell's work of 45 years ago, laying out these principles. And it's been an important way of thinking about cancers for many years. But I'd argue that where it really took a major leap forward was in the genomic era, when this understanding of cancer as a disease of evolution became a really crucial principle for being able to make sense of big data in the cancer field. And cancer is one of the hardest big data problems in the biomedical domain, I would argue, because it involves highly mutable cells. You have to keep track of an entire genome, and mutations anywhere in that genome might be relevant to tumor evolution. Different cells in a single tumor will have different combinations of mutations. Different tumors will have different mutations and different pathways through a selective landscape. And all of this needs to be interpreted using large, noisy, heterogeneous data sets, as well as in terms of very complicated biology that we don't fully understand. And evolution became a window through which we could make sense of a lot of this complexity and be able to extract some meaningful information from tumor genomic data sets that are so hard to interpret because of all of this complexity. And one of the key ways that happened was through the development of the field of tumor phylogenetics, which is basically just the idea of bringing phylogenetics or evolutionary tree inference to cancers. So being able to build evolutionary trees of tumors. Let me get a pointer here. So build evolutionary trees of tumors where we can take tumor cells, build a tree of those cells, identifying how they separate into different lineages, what mutations lead to this separation, and how that ultimately underlies the progression of a cancer. So all of that is something that's been studied for quite a while at this point, and there are now many variations on tumor phylogenetics. I want to briefly tell you a bit about this before I get to actually the interesting machine learning here. So my own group got into this field about 15 years ago now, working with actually pre-genomic kinds of technologies, in particular a technique called fluorescence in situ hybridization, or FISH, which basically just means fluorescently labeling pieces of DNA in a genome. And this turns out to be a useful thing to do in cancer studies, because one of the ways, maybe the main way cancers adapt selectively, is by gaining or losing copies of pieces of their genomes. FISH gives you a way of profiling this for a few pieces that you think are important, giving you markers that you can use to do phylogenetic studies, build evolutionary trees of cancer cells, and start to be able to tell these stories of how single tumors are evolving. Most of the field really took off, though, when it became possible to do whole genome sequences on cancers. So this is an example of a later method, really a set of methods that my group and many others have worked on, bringing these techniques initially to bulk genomic data, which essentially means genomic data on large populations of cells. That might be an entire tumor or a region of a tumor. This ends up creating a way of being able to see a lot more than you can see in those pre-genomic studies. In principle, see an unbiased view of all the mutation in an entire genome, but introducing another complexity that you have to deconvolve contributions from many different cells in each sequence analysis. This is typically posed as some sort of matrix factorization, but gives another way to identify a set of inferred cells in a tumor and how those might be related by a phylogeny that then gives you insight into what's functionally happening in tumor evolution. The field took another major leap forward with the work of Navin et al., who introduced single-cell sequencing to cancer studies. With single-cell sequencing, it became possible to get around the limitations of having to deconvolve data, which is a very noisy, low-resolution process, and became possible to look at tumor evolution at a single-cell level, looking at, in this case, these are profiles of copy number variation in a few different single cells, and get markers across an entire genome that could then be used to build yet other ways of constructing tumor phylogenies. Many of the most interesting methods today are multi-omic methods. This is an example of yet another method that's combining single-cell bulk sequence and FISH into a single inference by putting together a loss function that basically says that we want to infer cell populations, we want to infer how they're distributed over a tumor, we want to infer phylogenies among them, and we want to do this in a way that is consistent with how we think the biology works, as well as with all of our different data sources. And multi-omics is a lot of where the interesting action is in cancer studies. And there's a lot you can do even without looking directly at the DNA. So you can build tumor phylogenies from RNA, for example. And this is yet another example of some work of ours. As with all of these things, we are far from the only group working on this, but work of an example of an RNA sequence phylogenies from our method, in which we are bringing in knowledge of how RNA gives us information about activity of different networks and pathways in a tumor. Just as an aside, that this could probably be a whole talk about knowledge-guided machine learning, the different ways we bring in knowledge about pathways we know to be important in cancers, gene modules and pathways we presume to exist, but don't assume we know about in advance, giving yet another way of building phylogenies and finding out how those are reflected in functional activity of tumor cells or populations of tumor and non-tumor cells within a cancer. And that is just to illustrate that there are many ways of doing this, each of which has complementary advantages for learning about phylogenies. Now, what I've told you about is just a few examples of phylogenies from my own lab's work, mostly because it's easier to talk about and not worry about asking for permission to use figures from my own work, but we are one small piece of a huge field of tumor phylogenetics that is active today. This, just to give you a taste of that, is a table I put together actually about a year ago now, listing just some of the space of DNA sequence-based phylogeny methods that are out there. There are many for different combinations of data types, different kinds of genetic variation we care about, single nucleotide changes, copy number changes in the genome, structural variations like translocations, rearrangements, inversions, and so on. And these are just a small fraction of the methods that were available a year ago when I put this together. It's an even smaller fraction of the methods available right now as I'm speaking to you. The important point is that there are a ton of methods out there for many combinations of data, many ways of looking at this, many ways of saying what it is we're trying to track in tumor evolution or how we propose to do that. And I say that by way of background because it's really leading to the main thing I wanted to talk about today, which has to do with what we learn from tumor phylogenies. What knowledge do they actually encode that is useful for us for making practical predictions about cancers? Now, when we, and I think most other people get into this field, we had maybe a naive optimism that what we would learn from tumor phylogenies is a window into the story of every particular tumor. We would be able to look at a cancer, we would be able to tell the story of how that cancer developed, what mutations were occurring when in the cancer, what evolutionary problems those were solving, suggesting how this overcame an evolutionary landscape to get to, let's say, tumor growth, to get to metastasis, and then be able to back out from there, how we could develop better diagnostics, better targets for therapeutics, and so forth, and treat maybe that particular patient, more likely patients like that one in the future. And I call this naive because that idea does not work out at all. One of the things you learn very quickly when you work in tumor phylogenies is that you learn absolutely nothing from looking at a single tumor phylogeny. And the reason for this relates to something I told you about back at the beginning of the talk. We believe in most cases cancers begin with the acquisition of some sort of hypermutability, very rapid acquisition of mutations. And as a result of this, most of what is happening in tumor evolution has nothing to do with the functional adaptation of the tumor. This is something well known from work of others, especially Christina Curtis's group in what's called the Big Bang model of tumor evolution. But essentially, tumor evolution is a highly stochastic process. Most of what is happening in evolution of one tumor is just random noise, and you can't tell by looking at one tumor phylogeny what is meaningful and what is random chance in that tumor evolution. Fortunately, though, that doesn't mean that there was no point in looking at this. When you start looking at large numbers of phylogenies, you do discover that there is actionable information in them. And that also is something that became apparent fairly early in these studies. This is just an example of a very early version of this from our work. This one was almost a decade ago at this point, using some of those fish phylogenies to try to ask, what can we predict about progression of a cancer? In this case, the particular question that we were trying to ask is, can we look at primary cervical cancers, so the initial tumor that a doctor sees in a clinic, and identify the cancers that will progress to metastasis? And the short answer was that if we take information from the raw data that goes into building these phylogenies, basically different ways of slicing up the raw count data that we use to build the phylogenies, we do no better than chance in producing predictors of whether we're looking at a metastatic or a non-metastatic cancer. But if we look at this through the window of tumor evolution, so we build our phylogenies, we use those to extract features that we can derive from the phylogenies in different ways. So things that tell us about propensities to mutate by particular kinds of mutations, propensities for overall mutability in the tumor, propensities to evolve in, let's say, ways that promote more genetic heterogeneity or less. There's a lot of information in that that lets us make accurate predictors of which tumors go on to metastasize and which don't. And I'm not going to go into many years of work that followed up on that, but it basically showed that as we build better and better models of tumor evolution, these predictors become more and more powerful. Essentially, there's information in the evolution of these tumors that is ultimately encoded in the raw data, but you have to bring in the knowledge that you're working in an evolutionary system, the expert knowledge of how to extract information about the evolutionary process to make use of that. And that was a very general point that came up again and again in our studies in this field and others over the years. And that brings me to a summary slide I put together here that's trying to summarize a very large body of work over many years, but just to get to the point that's really underlying the main thing I wanted to talk to you about today. And that is summarized as follows. There are lots of ways of building tumor phylogenies, and quite broadly across these ways of building tumor phylogenies, we observe variation in the evolutionary processes of these tumors. Different tumors have different propensities to evolve in different ways. There are different hypermutability phenotypes. Some tumors have higher mutation rates than others. Some have higher rates of point mutations. Some have higher rates of copy number mutations. There are different subclasses in each of those. Some have higher rates of one kind of point mutation versus another, one kind of structural variation versus another. And this is something that shows up every way we look at tumor evolution, every kind of cancer we look at. Not only that, but that variation in mutational phenotype, so how a tumor is evolving, is quite broadly predictive of progression outcomes in cancers. Again, in our work and the work of many others, there are many ways we've asked this question on different cancers, different kinds of progression phenotypes, different phylogenies, different data sources, and it comes across quite broadly that how a tumor is evolving gives you predictive power for how it's going to progress in the future. And that's really the key takeaway of everything I've talked about so far, which is leading into the main point I actually wanted to get to, and the main thing I wanted to discuss with you over the remainder of this talk, which is the real question we want to ask here, which is how important is that knowledge? In terms of evolutionary theory, evolution has two pieces. It has drift, or the generation of genetic diversity, and it has selection. So picking diversity that gives an advantage of growth, or in some other way, an advantage to the thing we're looking at. In this case, the thing we're looking at being tumor cells. And in most studies of phylogenetics, most studies of evolution, indeed most studies of cancer evolution, the focus is largely on selection. We're largely looking in the cancer sense at driver genes, so genes that are believed to have some sort of functional advantage for the cancer. And in most studies of phylogenetics, drift is generally assumed to be a generic background process. It's something we're usually assuming is not a variable that we're looking at in our process. It's something that is just sort of happening in the background and is giving us a window into selection. There's certainly exceptions to that in both species and tumor evolution, but by and large, that is how we look at these things. And I would argue, based on the work we had seen so far, that in cancers, variability and drift is a very important piece of the puzzle of understanding cancers, and cancers give us a window into how that is occurring. So we want to look at this question of specifically how important is drift in cancers? How important is the background process for generating mutability versus the specific spectrum of mutations that occur in any particular cancer? And when I say how important, what I mean is how much do they control the risk of cancers moving to later progression stages? And I'd argue that that's an important question for a couple of reasons. One of the reasons that's an important question is, of course, that we study these things because we want to help patients. We want to save or improve patient lives. And I would argue that it's quite important if drift is a source of a large fraction of the risk, because we believe that variabilities in the drift process are a very early step in tumor evolution. So we would believe that in most cases, the acquisition of a hypermutability phenotype is something that is occurring years before a cancer is visible, really years before it even is a cancer. And that most likely when you see a tumor, you're looking at something that has spent several years, maybe many years, silently acquiring high rates of mutability before there's any visible phenotype of tumor growth. So if drift is important, it's suggesting that there's actionable information very early in tumor evolution, even before there is a cancer present. And we'd argue that that's something that we would want to know about. I'd argue this is also an important question for basic research into well, not just cancers, but into evolution. So evolution has been a crucial guiding principle for understanding cancers, but I think it's less appreciated outside the cancer genomics field that cancers are also a great system for understanding evolution and can teach us some things that are very hard to learn in other contexts in evolutionary theory. Cancers are an unusual model system in evolution in that we get to observe millions of examples of navigating essentially the same selective space multiple times. And we can start to see patterns of how that's done in different situations and understand something I think actually relates to what one of our later speakers, Bin He, is going to talk about, how reproducible is evolution. Cancers are a great system for asking that question. And this is an example of one of the unusual things about cancers where we have this variability in drift processes that's hard to observe in other natural systems. So all that is really setting up the question we wanted to ask here. And that's where this really becomes a talk more about machine learning, that we want to use machine learning as the window of asking this question: how important is the evolutionary process and variability in that process and underlying cancer risk? Our overall paradigm for asking this question is to build evolutionary trees of cancers from DNA sequence data and use these to extract sets of features that describe the evolutionary process. Some of these are features that are describing things like overall rates of mutability, overall rates of particular subkinds of mutability, as well as overall features such as tree shape statistics that can describe whether we're looking at a process that leads to a lot of heterogeneity or low amount and so forth. And then we're trying to ask, how important are these features? How much of our ability to predict comes from this kind of knowledge specifically? And we ask that in contrast to two other sources of knowledge. We have driver features. So driver features are features that tell us about mutations in particular genes that are important to cancer. So specific mutations. In the evolutionary features, we're abstracting away what particular genes are being hit. We're looking at overall processes, and the drivers, we're paying attention to the genes. Now, we would expect drivers to encode some of the information in the evolutionary features as well, because ultimately hypermutability phenotypes come about because of mutations in driver genes. So in principle, those variabilities in evolution are redundantly encoded in the driver genes, but there's also information about selection that's lacking from there. We also look at clinical data, so basically metadata on things like patient demographics, things a doctor could observe in a clinic, things a pathologist would observe examining a tumor sample or biopsy in their laboratory. And then we want to ask collectively, out of all this information, how much of the risk that we can predict comes from the evolutionary information. We ask this in a number of different variations using two data sources: the Cancer Genome Atlas that we heard about in the previous talk, and the International Cancer Genome Consortium, a large international consortium, both of which are providing large amounts of tumor data. And we asked this question in a few different variations. We looked at two kinds of sequence data, whole exome and whole genome. Whole genome is much better for asking this kind of question because we can't see a lot of kinds of mutability in whole exomes. A whole exome means just only looking at the protein coding regions effectively. But we have to look at whole exome as well because the cohorts for whole genome sequence are very small. We simply don't have enough samples with whole genome sequence. We wanted to look at these bigger whole exome cohorts. We looked at two cancers in this study, breast and lung cancer. And we looked at two outcomes: overall survival, which means how long does a person survive after they've been diagnosed with the cancer, and disease-free survival, which is how long until they either pass away or have a recurrence of their tumor. And that gave us a number of different variations for asking this question. Now, I don't have a lot of time, so I'm not going to go into great detail about the actual machine learning methods we use. Basically, we use a kind of heuristic feature selection based on stepwise forward discriminant analysis, combined with a specialized kind of machine learning for this sort of survival data known as Cox regression with L0 regularization. And if we want to use this, we basically have to show first that we can make predictions. This is just showing that we can indeed, using all of those features, make accurate predictions. This is in particular showing that those models give us a way of accurately stratifying patients into good and bad prognosis subgroups and getting a significant separation. We do a lot better with the less informative but large cohort whole exome sequence than the more informative but small cohort whole genome sequence. Some of those are kind of at the threshold of significance here, but we are at least able to build good classifiers. And that lets us start getting to the question of what we actually learn from these classifiers about the importance of particular feature types. We started by just asking for individual features, how important are they? And this is a way of asking this in terms of over many random replicates, how often does a feature show up as significant following the feature selection process? And the short answer is that features from all three of these classes show up as significant. This is the breast cancer whole exome sequence. We get evolutionary, driver, and clinical features showing up. In general, the clinical features are the best, which is what we would expect. Clinicians have done a good job of finding a feature set, but there are predictive features from all of these classes. And we see similar things when we look at the whole genome sequence, similar things when we look at the lung cancer, which I'm not showing here. Individual features actually drop in reproducibility from the evolutionary class when we go to whole genome sequence, which I think is a combination of more redundancy among the features when we have more of these features available, as well as the smaller cohort sizes, making it hard to get significance on ones that are kind of at the threshold there. So that's telling us that there is information in all of these feature classes. In particular, related to our question, there is information in the evolutionary features. But our question is hard to answer nonetheless, because there's a lot of redundancy among this information. And that's captured in this matrix of cross correlations, basically pairwise correlations between different features. This is showing in red highly correlated features. So the two axes of these matrices are just the feature sets aligned against one another. So the ones that pass the feature selection. And essentially what we see is a partition into a few submatrices. The clinical features pretty much partition into one submatrix with relatively low correlation internally and with the other feature sets, which is just suggesting that clinical features work well. Clinicians and clinical researchers have done a good job of finding a small number of things you want to check that give you a lot of relatively non-redundant information. We next see a couple of submatrices that turn out to correspond to the evolutionary features, and they pretty much partition into one group for the structural and copy number variation features, another for the point mutation features. There is substructure within each of these. It's harder to appreciate on this diagram, but to a first approximation, there are two axes of variability corresponding to those two kinds of mutability. And that's not so surprising in light of the prior literature. Driver features show up as their own submatrix, and we have a kind of weak mutual correlation among all of these submatrices. And we think that's largely occurring because of a kind of proxy effect, where if you have a highly mutable tumor, you will see a lot of driver mutations. And if you look at a tumor with a lot of driver mutations, more likely than not, you're seeing a highly mutable tumor. So that's why we think we're kind of getting largely this background pattern of mutability. And we see similar things in all of the other cases. And there is evidence for a lot more correlation structure here that we're not fully exploiting. I just show you an example of this by doing a dimensionality reduction, taking those evolutionary features into a two-dimensional space with t-SNE. And essentially what this is showing is how point clouds of the tumors correlate to the progression outcomes here. It at least looks like superficially there is a good correlation there in the breast cancers, maybe islands of good outcomes in spaces of largely bad outcomes. In the lung cancers, it almost looks like the opposite, islands of bad outcomes within spaces of good outcomes. But at least to a first approximation, it does appear that probably we're seeing these same two axes of features here, more or less point mutability and structural variation mutability. And there's a lot of information just in knowing where a tumor sits on those two axes. And that's not so surprising. It's been known for a long time. More mutable tumors have worse outcomes. Tumors that are mutable by what's called chromosome instability, so basically a propensity to have structural variations, have worse outcomes. And that ultimately lets us come to the real question we're trying to answer here, though, which is, among all those features, how important are the evolutionary ones? So we decided to quantify this in terms of hazard ratios. Essentially, how much information does a subclass of features give us to figure out someone's risk of developing a particular progression outcome in this kind of time series survival data. And essentially what we're able to find is that every class of features has significant predictive power. It depends exactly how you ask the question, how much predictive power there is, but evolutionary features have power, driver features have power. When you put them together to get what we call genomic features, which is just shoving the evolutionary and driver features together, you have more power. So each of those has predictive power, but each has some non-redundant predictive power, which is suggesting that we get something by looking at the mutations through the window of evolution. The clinical features are consistently the most powerful, but when we add genomic data on top of that, we get an extra boost of power beyond that. So again, there is more to the tumor than we can see from the clinical data. And we can use that to visualize an answer to our question, a kind of rough quantification of how important these features are, based on how much of the progression risk is contributed just by the evolutionary features. These circle plots are showing in orange the amount of hazard coming from the evolutionary features, in yellow the amount of extra hazard we get when we add the driver features beyond those. So effectively taking the drift in the orange features and adding information about selection on top of that. And then the red is bringing in all the kind of extrinsic factors beyond the genome that would be available to a clinician, things like patient age, demographics, pathology reports, and so on, which contains quite a lot of predictive power. And that lets us draw a very rough approximation of an answer here, that more or less the amount of importance we attach to the drift is the amount of orange in each of these circles. And very roughly, we'd say it's about 1/3. And let me emphasize here that I would not attach any importance to whether it's 1/3 or 1/4 or 2/3 or whatever. The important question here is really, is it roughly 0? Is it pretty close to 0? Is it pretty close to 1? Or is it something in between? And I would argue that this study is saying very clearly it's something in between. Drift is an important part of underlying progression risk. That is variability in drift processes, variability in somatic mutation mechanisms. It's not everything, but it's far from nothing. And again, I would argue that is a conclusion with important implications for cancer research and treatment, because it's suggesting that there may be actionable information long before we actually have a tumor readily apparent, if we can figure out how to get at that information reliably. I know time is short here. I think I really need to wrap up. So let me just say that one of the reasons I like to talk about this is because I think this study could be done so much better than we did it. There was a lot more to be done with bringing this to other phenotypes, other data types, other kinds of cancers with actionable progression questions. There's a lot more that could be done by bringing more sophisticated evolutionary theory to this problem, and a lot more that could be done by bringing more sophisticated machine learning, and especially better uses of knowledge in machine learning to this problem, including knowledge of networks and pathways that we know are important in these cancers, ways of designing machine learning models simply to use the knowledge that we know these cancers act through biological systems, that we know they act through tumor heterogeneity and evolution, and other ways of taking advantage of the structure of the data space, for example, through more sophisticated manifold learning ideas. So let me just close there by acknowledging the contributors to this work, especially my student, Yifeng Tao, who's lead author on a lot of what I described to you today. Yifeng is actually defending this afternoon, so I hope you enjoyed his work. We're really proud of him. Thanks to others in my group who contributed, and to work from my collaborators' labs, Lab of Chen Ma at CMU and Xinghua Liu at the University of Pittsburgh. Thanks to our funders, and thanks to all of you for your attention.
Thank you, Russell, for the great talk. Let's see the questions. Okay, I'm going to read the first question. Russell, have there been any comparative effectiveness studies comparing different tumor phylogenetic methods against one another given the same data?
Yes, that's something that comes up really most of the time when someone designs a new tumor phylogeny method. It's just expected that you'll benchmark it against other methods. It can be a challenge because different methods are constantly being designed that use a new kind of data, for example, and then there's nothing to directly compare it against. But that's something that we would routinely do when we develop a new method. We compare it as best we can to other methods and anyone else in the field doing that. I'm not sure if I could cite a particular review study where someone has just done that comparison more broadly. I'm sure they exist, but off the top of my head, I can't think of one.
Okay, thanks, Russell. The second question is, Russell, for the explained variation of progression risk, the genomic features appeared to be tested first, and then the clinical features. If the order were reversed, question mark.
Yeah, so you could reverse the order that we're doing that analysis, and it would really be asking a different question. Our concern here really was how much information do we get just from the evolutionary features, but then we would be getting that question, then the question of how much information do we get just from the genomic features. If we had done the clinical first, those have redundant information with the genomic features because they do include some assays for important genomic effects as well as other information. So it would really be asking a different question about how much we get just from the clinical data and how much does genomic data add on top of that. And you can kind of get that conclusion from the things I presented, but you would end up with a somewhat different answer that really the clinical data contains most of the information, but then genomics is giving you a boost beyond that. So it does allow you to improve your accuracy over current clinical metadata.
Okay, thanks, Russell. We're just, at the time, 11:30 for the next speaker. So thank you, Russell, for your effort and contribution to the session. And great talk. And great to see you.